Continue our session 2 nitrosamine session. The first talk in our afternoon session is from Pradnya Shahapure, and who is a team head for biopharmaceutics from some pharma industry. So I will introduce her presentation title. Her presentation title is PBPK Modeling-Based Evidence to Support BE Preservation Following Nitrosamine-Driven Reformulation of a Modified Release BCS Class 2 Oral Drug Products. Welcome, Pradnya. So she will be presenting her presentation online.
Thank you, Dr. Fang. First of all, I would like to thank FDA and CRCG for giving me the opportunity to present this work. I'll share my screen now. Yes, so I hope the screen is visible. I'll start with the presentation. The topic of my presentation today is PBPK modeling-based evidence to support bioequivalence preservation of a reformulated BCS Class 2 modified release oral drug product. A quick disclaimer, I'll start with that. Views expressed in this presentation are my personal opinions and they're not endorsed by Sun Pharma. In today's presentation, I'll give a brief overview of the regulatory framework for nitrosamine-driven reformulations. Then I will present the case study of a delayed release capsule of BCS Class 2 molecule and discuss the key learnings from the case study. As we are aware, the guidance on control of nitrosamine impurities describes NDSRIs, which are unique to each API. The options that are suggested for NDSRI risk mitigation include screening of excipients and addition of antioxidants or pH modifiers. Addition of the excipient is a SUPAC level 3 change. It requires the demonstration of bioequivalence of the reformulated product with the pre-change product or the RLD. Depending upon the BCS class and the type of formulation, various strategies are suggested for the approval of reformulated products. These include alternate BE approach through multimedia dissolution, bridging studies wherever applicable. Along with that, we have in vivo bioequivalence study, in vitro in vivo correlations, or PBPK modeling as the other suggested approaches. I'll now take you through the case study of an approved and commercialized delayed release capsule formulation of a BCS Class 2 drug, in which N-nitroso impurity was identified. The reformulation strategy for nitrosamine risk mitigation included the addition of ascorbic acid at the drug layering step. Amount of ascorbic acid was less than 10 mg per capsule, and quantity of sugar spheres was reduced to maintain the final capsule weight intact. A seal coating separated the drug core from the enteric coating. As a result, ascorbic acid did not come into direct contact with the enteric coating. Integrity of the enteric coating was not compromised in the reformulated product. The prior approval supplement was submitted with multimedia dissolution data for approval of the reformulated product. We received a regulatory query. The regulatory gave us a major deficiency, citing that the information provided to support the alternate method was inadequate for demonstrating bioequivalence. To support the proposed change in the prior approval supplement, the agency recommended that we conduct an in vivo fasting bioequivalence study on the reformulated product, comparing it with the RLD. Alternatively, the agency also suggested a biowaiver approach to provide evidence that the antioxidant, that is ascorbic acid, did not have any impact on the absorption of the drug as compared to the approved test product. So we decided to adopt the PBPK modeling strategy to support a biowaiver. During PBPK modeling, we integrated the physicochemical properties, formulation parameters, enzyme kinetics, physiological parameters for model development. The key parameters that were optimized were the effective permeability, fraction of drug unbound in the enterocytes, and enzyme kinetics. We also performed the parametric sensitivity analysis to identify the effects of these parameters on the model. Then we conducted an internal validation using dataset of an in-house study, and the prediction errors were found to be well within 15% for both Cmax and AUC. The model was thus internally validated. This validated model was then applied for demonstrating the bioequivalence between the reformulated product and the RLD by conducting virtual bioequivalence trials. The model demonstrated bioequivalence as the T/R ratios were found to be well within 80 to 125%. This model was also used for demonstrating the risk of increased permeability due to the addition of ascorbic acid. It successfully showed that even with 10% increased permeability, the bioequivalence was not compromised. These are the results of the PBPK modeling. This was the original study that was used for model development. And these are the T/R ratios, the VBE trials for the different, this is for the pre-change batch, for the reformulated batch, and for the batch with increased permeability. So we submitted the prior approval supplement using this PBPK modeling approach to demonstrate bioequivalence. We again got a query from the regulatory in which the agency mentioned that our model development strategy was reasonable, but wanted us or recommended us to go ahead and prove three things. First, evaluate the impact of a wider P-effective change on the bioequivalence during virtual bioequivalence analysis. Second, conduct external validation for the PBPK model using independent data sets. And thirdly, justify the sensitivity of the model for detecting non-bioequivalent scenarios. So for evaluating the impact of the wider P-effective change, we went back and looked at the PSA, and in this we found that the permeability had a linear impact on the Cmax, but it was to a lesser extent on the AUC. Accordingly, the model showed non-bioequivalence when the P-effective value was changed by more than 30% to that of the optimized value. So here we can see that with values of 1.89 and 4.05 for the effective permeability, we found non-bioequivalence. We conducted external validation using dataset of another DR formulation, and the model showed bioequivalence for all the 5 VBE trials conducted on this. To demonstrate the sensitivity of the model to non-bioequivalent scenarios, we used dissolution profiles with varying dissolution rates. Sensitivity of the model was challenged using slower dissolution profiles as the in vitro function. Here we can see the slower dissolution profile. Now, since this was a BCS class 2 drug, the rate of absorption is highly sensitive to dissolution kinetics. That is why with slower dissolution, we could see an impact on the Cmax, but a lesser impact was seen on the AUC. This is also seen in the virtual bioequivalence trials. The first trial conducted with 50% hypothetical release in 3.5 hours demonstrated non-bioequivalence for both Cmax as well as AUC. Whereas in the second trial, when the slower release was extended up to 7 hours, we found that the AUC was not as much impacted. It demonstrated bioequivalence, but non-bioequivalence was again seen for Cmax, which was actually expected. Thirdly, since permeability was not a limiting factor for the BCS class 2 drug, a faster release profile, hypothetical profile, showed that the bioequivalence was not impacted. So when we answered all the queries, we found that we got a successful pass approval by demonstrating the bioequivalence using this PBPK modeling approach. So these are the learnings and conclusions from the study. First, PBPK modeling is a reliable and scientifically robust biowaiver approach for establishing bioequivalence. Secondly, the reformulated test product successfully met the bioequivalence criteria. So we can conclude that addition of ascorbic acid, small amount of ascorbic acid, did not have any impact on the in vivo performance of the formulation. The addition of ascorbic acid at levels below 10 mg per dose did not alter the permeability, indicating that low levels of antioxidant do not adversely affect the drug absorption. Now, here is some food for thought. First, if reformulation interventions that mitigate risk do not alter the release performance or the in vivo exposure, should the reliance on in vivo BE studies for DR products be reconsidered? Secondly, can the alternative BE framework for in vitro dissolution currently accepted for IR formulations be scientifically and regulatorily extended to the DR dosage forms? Ultimately, our aim is to bring the drug products to ensure uninterrupted supply of the drug products to the patients. So simplification of the approval requirements will surely ensure that we achieve this objective. So with that thought, I will conclude my presentation. These are the acknowledgements. I thank the team back home, Dr. Lalit, Dr. Sudarshan, my boss, Dr. Arshad Khuru, Dr. Sumit Madan, the head of formulations, Dr. Swagata Sinha from my team, and Dr. Dipanjan and Dr. Dipanjan and Dr. Ramkrishna, the personnel who worked on this project. Thank you so much, everyone. And thank you, everyone, for your patient listening.
Thank you, Pradnya. So next, we will have Dr. Ming Guo from Office of Bioequivalence, OGD, FDA. And his talk would be analysis of ANDA submissions of bridging pre- and post-change nitrosamine impacted drug products and the regulatory experiences. Welcome, Dr. Guo.
Thank you, Dr. Fang, for the introduction. And my name is Ming Guo. I'll be talking about the ANDA submissions of bridging pre- and post-change nitrosamine impacted drug products and some case studies as well. And this is the disclaimer. This only represents my point of view, not of the agency. And the outline for today is give you some background of the nitrosamine drug substance-related impurities and the alternative BE approach principles, and some statistics, the numbers about ANDA submission over the years, as well as the control correspondence regarding reformulation of nitrosamine impacted drug products. And finally followed by some case studies and summary. All right, the nitrosamine drug substance-related impurity, NDSRIs, I would not read word by word in this slide, but in a sense, the NDSRI is a cohort of concern and can be originated from the APIs, excipients, and manufacturing process, or the combination of any of them. So in response to those root causes, generally the mitigation strategy has three aspects: the control of the API, control of the manufacturing process, and I'll be focusing on the third part, the control of the formulation or the reformulation, which further includes the addition of the antioxidant or pH modifier, and replace the problematic excipients with alternate excipients with lower levels of nitrite, or change the excipients, which will be exemplified in my case studies. All right, this is the statistics for the nitrosamine impacted reformulation on ANDA and correspondence control since 2018. As from there, it first draw the nitrosamine impurity first draw attention once it's found in the sartan-based medications. And over years, as you can see, the numbers of the related submissions been growing, almost doubled every year. But I want to underscore those are just, although seemingly they are not a huge number, but they are the submissions related to reformulation, which sort of a last resort of the applicant would do. And by far, the 2025 we have 34 applications or submissions or correspondence controls. And by the type of the drug class, the vast majority is still antidepressant, antihypertensive, and antidiabetics. Although the theme of the day is one of the themes of the day is oncology drug, but as far as I recall, we don't see reformulation product for the oncology drug products. This table highlights the BE approaches for the reformulation, nitrosamine-driven reformulation. More importantly, the alternative BE approach that is summarized from the nitrosamine impurity guidance. In short, the guidance favored the IR product, immediate release product, for the BCS 123, if you're using the 4 FDA researched antioxidants and the pH modifiers less than 10 milligrams per dose. And you can waive the in vivo BE studies in lieu of the in vivo BE studies with the comparative dissolution study on the pre and post change products in QC and multimedia dissolution conditions. The more you deviate from that, the more supporting data you need in order to support your changes. And generally, the MR products is not recommended for the alternative BE approach. And of course, this is in the assumption of no major manufacturing changes as well. And although the guidance, nitrosamine impurity guidance, gave us some alternative BE approach to lessen the burden of in vivo BE studies, but the reality is not always a clear cut in the situation defining the guidance. So that's where this case study is going to shine when the mitigation strategy is more complicated than the scenarios in the guidance. How we evaluate those mitigation strategy, if they are acceptable or not. So I give you 3 case studies, or more of maybe 4 if you break down the second with 2 cases. So the first one is a hypothetical case study, is IR product with 2 strains, high and low. And since the nitrosamine issue has been identified, the mitigation strategy finally come down to a reformulation by the applicant. So the reformulation is by adding by changing the excipient A grade with lower nitrite content. The excipient A, which typically is a filler or diluent, and in both strains. But more changes has been made to the lower strains because, as you can see, the not only excipient A but also B and C has been, and also the film coating has been added to the lower strains. As a result, the tablet weight of the lower strains has been significantly increased. And the applicant proposed to submit the comparative dissolution on the pre and post change products on the QC and the multimedia dissolution condition to waive the in vivo BE study. So how are you going to evaluate it? So this is a mixed effect, right? So there some of the changes are in favor of a waiver, and some are not. For example, as you can see, the lower strains has been gone through a major change. Many excipient increased. So it's a level 3 by the Supac IR because there's no antioxidant involved here. So we by default fall back to the Supac IR guidance. And also since the increase in the many excipients is no longer proportional, no longer can maintain proportionality for the post-change lower strains to the approved high strains. So those are not in favor of a waiver. But on the other side, the high strains is just a level 2, since it's a technical grade change. And also in terms of the excipient safety, because many of the excipient being increased, we have to evaluate it still under the inactive ingredient limits of the approved drug products in the same context of use. In this case, it's still under that limit, so we don't have the concern of the excipient safety. Also, the dissolution data provided by the applicant prove that it has a very rapid dissolution in all the dissolution conditions. And also you can think of if there's other waiver pathway, for example, the BCS, because the applicant demonstrated the high solubility for this one without further demonstration of the permeability. So we fall back to BCS3 putative. But again, mostly the solid dosage form doesn't really Q1, Q2 to the RRD, so you cannot go by the BCS3 based biowaiver. So as you can see, some of the in favor of waiver, some are not. Eventually, are they still eligible for a waiver? And so in the end, our conclusion is the recommendation is the in vivo study can be waived based on the totality of evidence. I know you heard of totality of evidence many times. The basic rationale we know is IR product is relatively low risk and high solubility across all the physiological pH ranges, and the very rapid dissolution has been demonstrated. But the key issue is the increased excipient in the lower strains. Would they have any impact on the bioavailability? So that goes to the scientific justification. For the reason of the credentiality, I cannot disclose what exactly the justifications are. But at least I can tell you is this can include like available RRD information and some literature or some your in-house data. But everything, the take-home message is in such unique cases, not clearly defined in the guidance, the agency is open to discussion. But at least you have to initiate this discussion to provide a starting point. What can be assessed. So those are the scientific justification. It can also differ from one drug to another or one applicant to the other. So overall, again, this is finally as come out as a good result of the in vivo B study. So next one, I want to compare side by side, which is a modified release drug product. More specifically, it's a delayed release drug product. So 2A, 2B, the same product, right? Just by different applicants, and they also have the nitrosamine impurity issues, and the mitigation strategy is very similar. So they both added the antioxidant less than the recommended antioxidant in the guidance, less than 10 milligram per dose, and as well as the pH modifier in the IR layer. And the delay release structure has already been exemplified in the last talk. I wouldn't go in detail, but rather it's just a DR layer wrapped around the IR layer. So a relatively simpler formulation compared to other modified release drug products. But the only difference is the manufacturing changes in 2B. Everything else is the same. But 2B, the manufacturing changes, we have to further examine if that's gonna have any impact on the release control, the delay release coding, right? So the 2A, in the 2A, the applicant proposed to conduct permeability study to support this antioxidant not going to affect the absorption of the drug, and also the in vitro dissolution data typically. And the 2B, the applicant only proposed in vitro dissolution data to waive the in vivo BE studies. So our evaluation, because it's MR product, is generally not recommended. The alternative BE approach not recommended in the nitrosamine impurity guidance. And also then we go back to look at the SUPAC IMR. Both apparently are level 3 changes based on SUPAC IMR. And but if you go back to the previous slide, oops. Again, the change is only being made to the IR layer, not the entire coating. So we believe the release control layers has not been compromised. They still maintain the integrity. And also the applicant provided 2A, for the applicant 2A, provide the data to support that the API is highly soluble. Although no further information on the permeability, so we fall back to BCS3 putative substance. So that way we can think of if we can still leverage the nitrosamine guidance to treat it as an IR product. So that way, in the end, we consider the in vitro dissolution study with the pre and post change and in multimedia dissolution could suffice bridging the pre and post change products. But for the 2B, this is a evaluation both OGD and OPQ, right? Because they involve the manufacturing changes on the CMC level. This is just a CVE or somehow similar to a level 2 changes. But this change involving the enteric coating. So we have a concern if the release layer has been compromised or changed, the integrity has been changed. So we need more data to address our concern. So you cannot just use the in vitro dissolution to solely as the supporting data. So that's why in the recommendations for the 2A, we consider there's no in vivo BE study required, and the permeability study is not required either. And you can just submit the comparative dissolution data. And for the 2B, because of the changes involving the entire layer, so generally the 2 pathways. The first one on the top is just the standard BE approach. You almost like you start over the BE study all over again. Or again, if you still want to go to the in vitro pathway, you submit your comparative dissolution data, plus you have to prove this reformulated product doesn't have any impact in the absorption. The data can, the supporting data can be PBPK or any other studies you think is appropriate. And of course, for most of the MR product, there's a additional in vitro testing to ensure the drug release profile. So those studies you still have to perform. All right, the case 3, again, it's IR products with multiple strains. And also apparently the nitrosamine issue has been identified. The mitigation strategy involving the addition of the antioxidant less than 10 milligram per dose, and to compensate, maintain the tablet weight, excipient A has been reduced. But since it's only small amount, so overall the proportionality has been maintained across all the strains, and no other changes, particularly the manufacturing changes, being found. And the applicant proposed to leverage the comparative dissolution data on the QC and multimedia condition to support this change. All right. So this is how we evaluate the mitigation strategy. So again, although it's an IR product, but since the applicant cannot really demonstrate the high solubility of the API, according to the BCS M9 guidance and also the nitrosamine guidance, we fall back to consider it as a BCS4 class. Then it's not really qualified for a alternative BE approach, although on the other aspects, seem to be all good, like less than 10 milligram per dose and proportionality in the formulation. Therefore, the applicant's proposal to just have the comparative dissolution data would not be able to support this change. So our recommendation is, of course, because the antioxidant is a new excipient, we have to evaluate the safety to make sure it's under the IAG limits. And also we recommend in vivo BE studies on the higher strains and the comparative dissolution data for the other lower strains. Or alternatively, you can provide evidence the antioxidant wouldn't have impact on the absorption, drug absorption, bioavailability. So in summary, as you can see the statistics in the first few slides, so the nitrosamine impact and the submission have been increased over the years. I don't know over the next few years it would be maintain the same level or not, we'll see. And also the nitrosamine guidance offer us some opportunity of the alternative BE approach, but that's not the only guidance we should be looking at. we also have a combination of all the SUPAC and MNIC guidance all together to examine case by case. And the particular mitigation strategies often come with very complicated cases. So there's no one fit all scenario. You can see this definitely you can go by alternative. The other one had to go by in favor BE. So the data need to support the reformulation. It's really by different drug products, by different applicant on your drug specific and the scientific factors. And as you can see, we've been talking this over and over again. As soon as you see your case is not really clear defined in the guidance, it's highly recommended to engage with the agency through the through the meeting request or the control correspondence, see if we would agree on your bridging strategies to save the time on your drug development. And with that, I'm gonna thank the management of the OV Office of Bioequivalence, and most importantly, I'm just delivering the message and give the kudos to all the nitrosamine working group members, also the assessment team who reviewed those cases. Those are the real heroes behind the scene. Thank you.
Thank you, Dr. Guo. Now I would like to invite all the speakers and also the additional panelists to sit coming up at the stage, and we will start the panel discussion. And we have 3 industry panelists online. I think, yeah, they will participate in the panel discussion as well. And when they're coming to the stage, I would like to introduce 2 additional panelists, Dr. Dongmei Lu, who is the Associate Director for DTP from Office Research Standard and FDA. And another panelist is Dr. Dan Snider, who is the head of the Global R&D and the Technical Quality from Viatris. So right now we would like to first take the questions from the audience. Then we will take the online questions screened by our SME. And then we will see if we have time, we'll have some seeding questions to discuss. When you come to the mic, please direct your question to the panelist who you want to ask that question. Yeah, Dr. Pony.
Dr. Robnik online? Not sure.
Not.
He is okay.
Yeah. Here he is. Yeah.
Yeah. Thanks for your presentation, Dr. Robnik. So in case 2, that is, you added cysteine intragranularly in the dry state, you say, as an approach to mitigate nitrosamine formation. Is there any reason why you added? I guess more generally, was there? Did it really matter how you added the nitrosamine to the formulation? Was it process dependent? Did it make any big difference in the degree of mitigation?
Yeah, it's a little bit hard to hear, or was a little bit hard to hear the question, but I think I understood it. So yeah, we tested extragranular addition, and we call it intragranular, but in fact it was a addition into the solution. So it was dissolved in the liquid and then sprayed on, used during the fluid bed granulation process. And the impact of this decision on the nitrosamine formation was quite significant. Several fold of a difference in nitrosamine level reduction was observed when we used the L-cysteine in a solubilized form.
Thank you.
Any other questions from the audience? Maybe we can get some online questions. Yeah. Dr. Paul will read it through the microphone.
So I have several questions from the online audience. This one is specifically to Dr. Yi-Hsien Cheng. It says, in identified low risk BCS class 4, do we still need to perform PEPK modeling?
Okay, thanks for the question. So the short answer is yes, but in a way, I will view the PPK modeling as a supportive evidence. So to prove that your drug product is really low risk, you need to have a certain supportive evidence to prove that it's really low risk. With PPK modeling, it provides a great platform that we can either perform the sensitivity analysis, as I did in my presentation, or we can either use the virtual BE simulations to demonstrate that the post-change versus the pre-change reformulation formulations would not impact the BE assessments. Thank you.
Thank you for the answer. And yeah, is there another question?
I take one question for addressed to Dr. Rajkumar Boddu. Can we use biorelevant media like passive or passive to justify PE waiver for BCS class 4 drugs?
I think this is for Dr. Boddu.
Yes, this was addressed towards Dr. Boddu.
Yeah.
Can we use
the question is like,
media?
so can we use the biorelevant media resolution for the BE risk assessment for the BCS class 4 molecules, especially reformulated product, right?
Yes.
Yes. So if at all, if you're getting the good discrimination in the biorelevant dissolution media, so we can use that dissolution profiles as a part of BE risk assessment. So I have mentioned clearly the BE risk assessment framework. So after reformulation, so we need to evaluate the both solubility as well as the permeability as well as dissolution. So we should evaluate all these factors because the BCS class 4 molecules, so it is a low soluble, low permeable, right? So hence we should evaluate the reformulated product to know the impact of solubility, permeability as well as dissolution. So coming to your point, so if in addition to those solubility and permeability, if at all if you're getting the good discrimination in the biorelevant dissolution, so we can use those profiles to predict the BE risk. And we can integrate those in vitro data with the in silico methods like PBPK model, and then we can evaluate the BE risk.
Thank you, Dr. Boddu. Any more online questions?
Yes.
Okay, yeah, please.
This was specific to Dr. Bo. You presented a case which has showed bioequivalence using PPK modeling with dissolution of 85% in 15 minutes with ascorbic acid. Would this conclude that this product applies to class 1 products?
May I ask which particular case is talking about?
The question did not mention specifically.
So I think probably this question is to Pradnya. So I think you presented a PBPK case to support waiving of the nitrosamine impacted reformulation. So that is that is BCS2 drug product, I remember. And then that question is for whether the PBPK approach can also be used for evaluating BCS1. I think that question is
from
interesting.
Audience, that's right.
Yeah, BCS1, actually the nitrosamine guidance already cover that situation.
Okay,
Yeah.
that's all.
Okay. Okay, thank you. Thank you, Dr. Paul. So is there any questions from the audience? Yeah. please.
Hi, thank you. I was wondering if anyone has any experience evaluating the impact of pH modifiers on, say, precipitation for any drug products where precipitation might be an issue, like a salt or amorphous form or anything like that? Because I've heard a lot about dissolution and permeability, but do you ever request any alternative dissolution tests or other types of tests that could look at potential precipitation? I guess this is for anyone who might have that experience.
I think I can take up this with that. Because in terms of the pH modifier in the reformulated drug product, and in general, FDA also conducted several research studies and indicate that really tiny amounts of the pH modifier in the reformulated drug product can effectively prevent the nitrosamine in the SRI. So the tiny amounts, that is the key point, because think about the drug product after oral administration, it will go into the stomach first, right? So pH modifier basically we refers to this, such as the sodium carbonate, and so in the stomach, basically it should already get neutralized. So we don't think that there will be high risk in terms of this addition of the pH modifier is going to have some impact in the GI absorption. So that is the rationale underneath.
Okay, thank you.
Thank you. Another question?
Thank you for the presentation from all the FDA members about the BCS subclassification that you're doing. It's amazing work, and I think we need to continue in that direction. I have two questions about that. First one is just how are you still using current BCS biowaiver guideline for classification into BCS 2 and 4, which means are you using bioavailability or permeability? And the second one is how do you see this knowledge evolving also into the other fields of waiving based on the BCS, where we don't have it's like here we had a lot of learnings about bioequivalence before there was a change, right? So how do you see the knowledge that we are gaining extrapolating now into the cases where we will not have that kind of knowledge and we'll do some changes in formulation?
Maybe Dr. Cheng, you can give a comment first.
Okay. Yeah. Thanks for your question. So actually, I would probably like to circle back on my talks that we can use the PBT model as a supportive evidence to support the potential reformulations. So if we can use the sufficiently validated PBT model to demonstrate that it is low risk to have non-PE results after reformulations, it will provide the rationale justifications that you can further use the PBT model to support the biowaivers. And for the proposed framework, actually, I would like to say that this is not rigorously designated threshold. I just propose here to open a discussion. We can further discuss on whether this the threshold based on the dose number and the permeability and the bioavailability, whether it's a reasonable threshold to classify into these four subcategories, and how we can use the PBT model to further support the biowaiver. That would be the main point that I would point out here. Thank you.
Any panel members want to add? If not, yeah, I think that, yeah, so the subcategories, yeah, we will have a small group discussion, see whether we can categorize it as a BCS 2-like or BCS 3-like or for BCS 4 drug products. We can have a further discussion in a small group discussion this afternoon. So then I think I will ask some seeding questions. This first question will be delivered to Dr. Boddu. What are the critical input parameters for PBPK model developed for BCS class 4 molecules to avoid repeat PE studies due to reformulated nitrosamine product?
Yeah, thanks for the question. So for any PBPK model development, the physicochemical, biopharmaceutical, formulation, and physiological inputs are very critical, and their accurate integration is required. So these key input parameters include, like the physicochemical properties includes the solubility across physiological pH and the pK, log P, molecular weight, and so on and so forth. So coming to the biopharmaceutical properties, input parameters like the dissolution behavior, precipitation tendency, and intestinal absorption kinetics and effective permeability. Coming to the physiological and disposition parameters, some gastrointestinal transit times, the regional GA physiology, enzyme transporter kinetics involvement, and the clearance pathways. We need to give those input parameters. Coming to for reformulated products developed to mitigate the nitrosamine risk, especially for the BCS class 4 drugs, the formulation specific inputs become especially critical. These includes the solubility and bioelement dissolution profiles and permeability of the modified modified formulation. So incorporating these formulation dependent inputs enables the PBPK model to mechanistically predict the impact of nitrosamine driven reformulation on in vivo performance and assess the BE risk without necessitating the repeat clinical BE studies.
Yeah, a follow-up question is, what is the model validation and the sensitivity analysis strategy for the PBPK modeling?
Generally, the validation strategy is essential for any kind of model. So to establish the confidence in the PBPK models used for the regulatory decision making, generally, typically the validation includes the external or internal validation. In the external validation, we can do the this step by using literature or any reference data. We can simulate the published plasma concentration time profile across different dose strengths or different release rates if it is available. And we can demonstrate the model's validation to capture the dose linearity and non-linearity or any formulation dependent absorption behavior. That is one kind of validation. It comes under external validation. So coming to the other kind of validation, that is internal validation, we can utilize the in-house data like the pilot or pivotal or any development we study, the data spanning both bioequivalent and non-bioequivalent outcomes. We can utilize these outcomes as a part of internal validation. Further, we can verify the model's discriminatory ability between formulations with known differences in the dissolution or release characteristics. Then we can validate the model. So overall, a PBPK model is considered validated when it consistently demonstrates the predictive ability and sensitivity to formulation or any other changes and robustness across scenarios relevant to the reformulated IR or MR product.
Thank you, Dr. Boddu. I think Dr. Yi-Hsien Cheng can also give some comment on the sensitivity analysis because you also present that in your presentation.
Sure. Thanks for questions. I will further add some points on how we can use the sensitivity analysis to support our findings. So here we did a sensitivity analysis on the permeability, excipient led permeability change and the dissolution change and how this impacted BE. So in our findings, we use the sensitivity analysis to actually build a set space. So we further demonstrate that if our reformulated drug products fall within this set space, we can further use the default PBPK model to demonstrate that this is a low risk after the reformulations.
Yeah, thank you, Dr. Cheng. Next question I will ask Dr. Nasrin. In your presentation, you presented the extending BCS3 antioxidant the research findings to BCS class 4 drugs. So could you please explain how you justify that? Because they do have a different absorption mechanism.
It's a very nice question. I think it's kind of summarized my presentation. So the first of all, I want to add that we are not saying that BCS4 drug is actually acting like BCS3. It's not actually a blanket extension we are proposing. We're saying that BCS4 drug is not like a big monster. We can subcategorize it. And then we are saying that there was an extensive research in our division, I think Dr. Dongmei here, that there was a, we did we seen that all the there were 10 antioxidant we tested on a BCS3 class drug, and we showed that none of them kind of changed the permeability of that drug. So that's why we are proposing that this BCS4 drug that's acting like BCS3 drug, that can be showing the same criteria. So, and then we kind of validated it using a case study where it kind of proved the point of using the BCS3 drug. I think we have actually a little bit of drawbacks that we don't have in our product. We just proved it in one product, but we'll keep compiling all the evidence and we'll try to validate it more in our modeling. But I guess I think I answered your question correctly. Thank you.
Yeah, thank you for giving the answer to that question. So I think that the next question will be given to Pradnya and Robnik and Cheng. Probably you can also comment in terms of the modified release products. When a nitrosamine-driven reformulation is limited to a minor quantitative substitution of a non-release controlling excipients with a highly soluble nitrite scavenger, what scientific considerations could support a BE's bridging approach? So first, I think I will ask Dr. Robnik. You can comment.
Yeah, when the change is intentionally low impact, and this is the most important consideration, that it's really low impact, then we would consider BA bridging as long as the evidence shows that there's no meaningful impact on drug release or the permeability. For instance, if the excipient to be changed is non-release controlling, so not involved in matrix formation, in erosion, swelling, or other release mechanism, then if the drug release performance is unchanged, meaning that comparative dissolution testing demonstrates similarity across different pH. So for instance, F2 is above 50 for all the media. And the drug permeability is unaffected, meaning that the excipients changes do not introduce any effect that could alter intestinal permeability or transport of the API. If all these checks are met, we'd expect no clinically meaningful shift in drug exposure.
Yeah. Thank you for the comment. And then Pradnya, if you can add something.
Yes, just one point that the amount of the scavenger, the dose that is, the quantity of the scavenger added, we should also look at that. It should be very small and it should not exceed the limit that has been specified in the guidance. Then in that case, we can consider a bridging approach. So overall, as Dr. Robnik has said, what we can summarize in one line and say that if the critical quality attributes of the formulation are not impacted by the addition of the excipient, then we can consider using a bridging approach. Thank you.
Yeah. Thank you. Thank you for comment. Yeah. Dr. Snider, yeah, if you have
any
comment.
Thank you for the opportunity to talk. The formulator has a lot of information available to them when you've already successfully developed, submitted, and had your product approved. You know what the dissolution profiles look like for your passing product. You know what the reference product looks like. You have the benefit of knowing all the trials that it took for you to get to to an approvable product in the first place. The changes that we're talking about are either going to be relatively minor or relatively major. And the formulator, as they're designing their follow-on product, has the opportunity to choose judiciously, right? You can choose whether or not to put in a hydrophilic or a hydrophobic sort of antioxidant. You can decide where you put it. And if you do it sequentially, stepwise, with a good level of product understanding and mechanistic understanding, then you can take the right decision. So as you build your follow-on formulation, you can build the necessary data to make your argument up front. Some will be very, very easy to manage, and some you may look at the data and say that you'd be more comfortable for the patient's sake to do a bio study. But you have that opportunity during the reformulation process to pick something that fits into your existing formula and mechanism of release so that you can choose wisely. And as long as the nitrosamines and the rest of the stability attributes achieve the effect that you anticipate, then you should be able to make a normal sort of model where passing dissolution across multiple pHs from a similarity perspective should be a relatively achievable submission.
Thank you. Thank you for the nice comment. I think in terms, I will ask a last question to Dr. Minguo and Dr. Dongmei Lu. Based on your like experiences of the ANDA review and other experiences, so for the nitrosamine-driven reformulation, what are the most common deficiencies that you see? And also, what are the trends or your comments in terms of successful versus unsuccessful approaches to demonstrate BE preservation?
All right, thank you. I'll go first. So although each individual deficiency can vary from one to another, but in general is that well, a lot of times we're in a disagreement with the applicant that their supporting data will not able to suffice the level of change. So just like Dr. Snider said, you have to understand your product, your process mechanistically and systematically analyze according to all the available guidance, which are in favor of the alternative B and which are not, and come up with an optimum approach. And also maybe not a very unpopular opinion is that although we are talking about other alternative B approach like modeling, PKP, PB, and IVIC, but we've seen applicants that, like for example, the last case, the case 4, I introduced is the IR product, but the BCS4. In our current practice, we still either recommend the in vivo B or PBPK study. But I've seen the applicant, they just go straight up to in vivo B for some of the product. Maybe it's easier rather than you going back and forth for your model validation. That's also time consuming, but maybe not the most cost effective way. So just factoring everything and come up with your best mitigation strategies. Yeah.
Thank you, Dr. Lu.
I think in terms of the second question, and Ming and I can provide some information. And what would be the best strategy seems really successful in terms of the reformulation, reformulated products BE. So I think there are several perspectives that we can consider. First is to try to engage with FDA as early as possible, because when you have some kind of a general idea about approaches, you want to propose for your study reformulated drug product, and to try to get FDA's advice, and either from control correspondence or request a meeting. And so in this way, you can buy your time, definitely. And the second point is that scientific justification. And in terms of this way, of course, your testing and your understanding of your product is very important. But at the same time, literature information and some really successful in-house examples, or some example you find from other seminar or presentation from other industry, if that's going to be helpful to your cases. And so definitely we already say that totality of the evidence will be the base for our FDA evaluation. And the third one, it is in terms of the enterprise thinking, and in terms of the cost, in terms of the time, which will be more important to you. So I think each applicant, if you want to have a justification and to give a more economic way of your product and your firm, this is a very important consideration for you. And Ming, do you want to add something?
Yeah, I think that's very important to have because all these cases in the stats, they are not just a single one round submission. Multiple correspondents being just go for one drug and eventually finalize as a PAS. So that's how important you have this engagement with the agency early in time.
Thank you so much. Thank you for all of the speakers for your wonderful presentations, and the panelists for your comments and discussion. Thank you. This concludes session 2. Now I would like to welcome Dr. Heather Boyce for kicking off the session 3. Thank you.
Good afternoon. It's my absolute delight and pleasure to co-moderate session 3 with Dr. Sivacharan Kollipara, who is also the Vice President and Head of Biopharmaceutics at Dr. Reddy's Laboratories. This session here we have today is specifically about discontinued RLDs and what happens when there's no reference standard available for applicants to test bioequivalence for their product. As Dr. Rob Leinberger alluded to earlier, this is one of the cutting edge agenda items that we're leading in the Office of Research and Standards in support with our colleagues across the FDA, Office of Biostatistics, and Office of Bioequivalence, to really carve out and create pathways for generic applicants to have generic products of specific drug products that have important public health utility. For instance, patient-centric dosage forms. If we have suitability petitions where there's no reference standard, you're going to hear a lot of different case examples and the overall story of how we're working through these problems at the agency. So this really is cutting edge. So I hope you all listen very intently and come with your great questions at the end of the session. So with that, I would like to introduce Dr. Suparna Mukherjee, who is the Director of Global Clinical Operations at Teva Pharmaceuticals. And she will give us the industry perspective on what it takes to develop generics when there is no RLD and no RS, and how we can overcome that barrier. So welcome.
Good afternoon, everyone. Thank you to CRCG and FDA for giving me the opportunity to present today. I'm Suparna Mukherjee, and I'm presenting on behalf of Teva Pharmaceuticals. So my topic is very central to generic drug development. It is what we need for any generic drug development for submission to the regulatory for approval, and how the structural barriers which get created when the reference listed drug or a reference standard is unavailable or difficult to obtain. I know the session is very focused to discontinued
RLD. However, from industry perspective, I think it's very important to highlight that not only the situations where we have a discontinued RLD, however, a situation where an RLD is unavailable or a reference standard is unavailable, how a generic industry faces challenge. So my talk will be focused on 5 such distinct scenarios where the industry encounters issues and how we can address few of the practical recommendations and how these challenges directly, hinders generic drug development or the competition and ultimately patients' access. So before we begin, just a disclaimer statement that the view which is presented today is totally based on my individual experience and does not necessarily represents company's official position. The examples and scenarios discussed are drawn from real-world observations, but may not capture the full breadth of industry experience. With that, to set the stage, let us remind ourselves why the reference-listed drug or reference standard is so central to generic drug development. As I was mentioning, we need these standards or the RLD to run the bioequivalence study, which are critical for the submission and approval of the product. Today, my discussions will involve basically 3 main pillars. First will be the scenarios where such withdrawal or unavailability of RLD or RS could create barriers for entry of the generic applicants. Few recommendations for each of these scenarios, followed by conclusions. Now, this slide highlights the 5 main scenarios which I will be talking today. The first one to begin with is discontinued RLD, which is the common scenario where RLD is discontinued and a new reference listed drug is being addressed to overcome that scenario. Second scenario is restricted access to the marketed RLD. Now, there is a marketed RLD available, however, it has been restricted from the brand in terms of the distribution. Third scenario is discontinued RLD with no designated RS. And I think this is the most challenging scenario which we will be discussing over this session. The fourth one is RLD and RS is active, but very infrequent commercial distribution. It can happen with few of the strengths or multiple strengths, but overall impacting the generic development. And then the fifth scenario, which is RLD or RS shifting to additional strength or a newer dosage form due to the market dynamics. So brand is coming up with a new strength and then or a new dosage form. And there is a shift in the RS or there is a shift in the entire market strategy on what product is being developed now. So now coming to the first scenario, that is discontinued RLD. When an RLD is discontinued, not because of the safety reasons, there is a well-established procedure from agency on how they then have a generic which has already been approved, labeled as a reference standard. Now, we all know that we need to do a bioequivalence study against this reference standard for proving bioequivalence and then submitting for the approval. And we thank the agency for this well-established process of designating RS. However, the question lies is the current scenario in which the RS which was getting designated is mostly based on the commercialization of the market leader of the current generic player or all the strengths availability. The open question here remains is, why can't all approved generics be designated as an RS since the market share is quite dynamic in nature? When we see over a period of time, what is today's market may not be tomorrow's leaders in terms of generics. So this is an open question I wanted to highlight here. The second thing which I would like to highlight in this case scenario is that when an original RLD is an RS, all generics players develop and run bioequivalence study against the original RS and prove bioequivalence. However, when this original RS shifted to a new RS, then the subsequent generic players are running bioequivalence to a new standard, which is bioequivalent to original product, but not identical. In such situations, sometimes we end up in a scenario where we see a bioequivalence drift or a BE shift, we call, which is not a theoretical challenge, but it's also backed by scientific evidences wherein we have seen that due to this shift, it adds up to the additional variability and challenges for a generic industry. So with that, I will move to the second scenario, which is again restricted access to the marketed RLD or an RS, which is quite critical for the generic drug development and submission. Now, again, I thank the agency for a well-established CREATE Act, which this pathway has taken care of majority of the challenges for these kind of product where the marketed RLD is through restricted access. The pathway is quite clear for non-REMS product where you submit with a sufficient quantities with a written request to the brand sets the offer, and it has to be within 14 days. The generic accepts the offer within 7 days, and then finally the sample is delivered within 31 days of the original request. For the non-REMS product also, pretty much the same process is followed. However, it has an additional step wherein you need to get requires a covered product authorizations from FDA and which followed a modified timelines. The point I would like to highlight here is that in spite of this CREATE Act, we often face some legal, operational, and practical challenges which continue to limit this access to RLD samples. Few of the legal and procedural barriers which include is this statutory requirements of the written request, a minute or a simple clerical errors can reset the timelines. As I said, for the REMS products, there is FDA involvement, which adds up to the 120-day CPA review period. There are sometimes disputes over what is a sufficient quantity based on what industry needs versus what a brand is offering. Ultimately, there is an enforcement required from the court in few instances, which again leads to a cost and a timing risk. Few of the operationals and practical barriers include that sometimes there is a claim that the product is available through wholesalers, but does not need to be a B suitable supply. We get the product which is often very short dated, near expiry, which creates a practical challenge. Pricing structures is another issue. Sometimes there is no feedback on the ancillary components, which is critical for the development and for our bioequivalence studies. And then there are logistic challenges. So the key takeaway here I would like to mention is that CREATE definitely is a critical pathway or a mechanism which helps in these kind of scenario. However, there is a structural and operational hurdles that remain and that can delay the timely generic development. Moving on to the scenario 3. Now, this is the most critical one in terms of today's discussion topic, wherein there is a discontinued RLD and there is no designated reference standard available because there is no generic in the market. The recent PSG outlining an alternative VE approaches is a very positive step towards this particular scenario, and we thank the agency for this. Now, I have listed few of the products wherein I have seen the updated PSG over the last year. Then one important limitations, however, I would like to highlight here, and we will see subsequently in the next slide as well, that most of these PSGs are currently outlining to the solids, but the dosage form, but I think there is a lot of work which needs to be done in a different dosage form, and how we create a framework for this alternative BE approach, and we move forward towards this addressing this challenging scenario. Now, since morning, we have heard a lot of time over totality of evidence. And I think for this kind of scenario, the thing which works could be the totality of evidence approach. And what I would like to highlight here is that when we say totality of evidence, it is not only the in vitro characterizations or a virtual BE or a PBPK modeling, but also I will take a step forward in terms of what we are seeing with the recent ICH M13 harmonization, the importance we can lay significantly on the use of a foreign reference. It need not to be the sole reference for proving bioequivalence. However, a foreign reference could be used as for exploratory studies when there are limited informations on the literature data. We can combine them in totality or using the analytical bridging being done between a generic, the available foreign reference product, and the literature data to come up with this simulation or a virtual BE standard. Moving on for the scenario 4. Again, this is a very subtle scenario. However, I feel it's very impactful, where RLD and RS is active. However, there is an infrequent distribution or a commercial distribution due to the market share. Sometimes in few of the dosage form, we have seen that we need multiple lots for establishing an in vitro BE studies, for example. And then there are sometimes it's needed to prove how the screening of multiple lots to understand the product variability. In such scenarios, when we don't get sufficient or it takes a longer time to get hold of multiple lots, the industry faces challenge. And then in such scenarios, our recommendations would be if a waiver pathway could be a firm can seek, wherein rather than using 3 lots, can we use 1 lot, or can we use a literature data to confirm rather than really using multiple lots for establishing the in vitro bioequivalence. Now, coming to the last scenario, scenario 5, again, this is a very different, I will say, a paradigm shift which happens from the brand side. There is a scenario where a situation where there is a change in the RLD strength from low to high. The expectations from agency is always to run bioequivalence study on the highest strength, or eventually we see a shift in the RS from the already available lower strength to higher strength. In such scenario, again, it's in terms of totality of evidence, along with actual in vivo BE study on a lower strengths, provided we have the formulation is proportional, we have a linear kinetics, we have an in vitro similarities. Can this be considered or as a recommendations for as a scientific surrogate, where in the actual RS or a high strength is not available? This guarantees a generic product to come to the market well in advance rather than waiting for the reference standard. Second situation is where a dosage form is changed completely, and there is a shift in terms of how brand is marketed. And in that instances, if a generic wants to develop an original RS, we have we have the challenges that the original RS may not be available. And if there is a new product which is currently leading the market, and if we decided to develop the new product or a new dosage form, then in that case, whether we can rely on the scientific evidence, how the brand has relied. Brand might have conducted bridging studies comparing the original developed product with the new dosage forms. Can a generic can rely on such alternative BE routes against the original dosage form, and again establishing all other supporting evidences, and in terms of totality of evidence, submit and application for approvability. So this is my conclusion slide, and as we have discussed the 5 main scenarios where we have seen that there is limited or discontinued RLD or limited RLD supply, or a reference standard is unavailable. Few of my recommendations will include, first is designate approved generics as an RS, like as I highlighted, the possibility of evaluating a different scenario in terms of what can be used as an RS rather than using the market leader. Acceptability of lower strength for bioequivalence as and when possible in terms of totality of evidence. Alternative BE pathways wherein we can establish bioequivalence against a different dosage form, and then relying on the literature, and then coming up with a totality of evidence in terms of establishing bioequivalence to the actual product where the RS is not available. And then waiver pathway for the multiple lots, as I highlighted, for few of the dosage form, whether when multiple lots are needed, and it's very difficult to procure multiple lots from the market from generic perspective. Ultimately, we thank the agency that there are several steps is currently been taken, and which is a very positive step towards this scenario to bring the generics accessibility to the patients. However, I feel there is more such structure engagements and discussions between different stakeholders need to be organized, and multiple scenarios need to be highlighted where there could be collaborations between the agency in industry, and these situations can be discussed and addressed. And before I conclude, I would really like to thank the management in Teva Pharmaceuticals, my peers, and definitely my supervisors, Elizabeth Rody and Thomas, who has really helped me putting together this presentation. And thank you for your time, and thanks for listening.
Thank you, Suparna, for that very well-articulated overview and providing some examples of opportunities for the agency and industry to focus on problem solving together. Our next talk will be by Dr. Hai-Lim Lim, who is a pharmacologist in the Division of Therapeutic Performance 2 in the Office of Research and Standards at the FDA. And Dr. Hai-Lim will talk about a very specific case regarding suitability petitions, which have gained more momentum with GDUFA 3, and they become more relevant. So she will discuss what happens when we have an approved suitability petition and what she is proactively doing in her work at the FDA to provide those pathways. So welcome, Dr. Hai-Lim Lim.
All right, good afternoon, everyone. My name is Hai-Lim. Thanks for a nice introduction, Heather. So today, as you can see in the agenda, this session 3 is really about discontinued RLD and RS. My talk fits with the theme from slightly different angle because suitability petition the applicants face similar challenges where the applicant wants to develop generic products, but there's no clear competitor to use. So in this talk, after my talk, I hope you guys walk away understanding our recent proactive efforts to provide clear recommendation in our PSG for B studies. This is the disclaimer that is not view of FDA, but for mine. All right, let's understand what's the suitability petitions first. So suitability petitions is the regulatory mechanisms where applicants submit the request to FDA to seek approval for ANDA that differs from the RLD in certain ways. The differences include the dosage form change, like from tablet to ODT, also the strengths, like new higher or lower strengths, and then route of administration, intramuscular to subcutaneous injection, and rarely one active ingredient change in fixed combinations as well. The goal of this suitability is to ensure that the generic drug is still safe and effective, even with these differences. The basis of submission of the first petition ANDA is the RLD identified in the petition and the approved suitability petition. Under GDUFA 3, there's an impact on number of suitability petition submissions. So under GDUFA 3 commitment, there's a commitment to review the suitability petition submitted on or after October 2024 within 6 months. The performance goal increases percentage with goal of 90% reviewed within 6 months in FY 2027. So as you can see in this graph on the right, like the red GDUFA 3, there's a dramatic increase, and that aligns with the GDUFA 3 commitment. And this shows industry response to this improved review timeline for the suitability petition submission. And let's look at the suitability petition landscape, or what changes are requested. So these are the petition basis distribution FY 1984 to 2025. So as you can see in the pie chart, the orange, the most commonly requested is the strengths, and next is dosage form, the blue, and maybe the both dosage form and the strengths. And in later my talk, I'm gonna share the PSG development that address those most petition type, like the strengths and then the dosage form. So here, this slide describes the challenges of PSG development for drug products approved via suitability petition. So for the challenges, for traditional generics, there is pharmaceutical equivalent product, and then there's clear competitor for B studies, and there's established study designs in PSGs. But for suitability petition-based products, there's no approved competitor in new dosage form or strengths in the market yet. And there's uncertainty about the study design, and there's limited guidance on this. Also, from the industry, we receive many of the control correspondence, and then the central question is, how do we design these studies for petition products when there's no approved competitor? The example of the question is, can we use a 2-arm or 3-arm study for orally disintegrating tablets like ODT? Do we need fasting and fat studies, or can we use multiple units of the RLD for higher strengths? And now here comes to our new approach, proactive PSG development. So traditionally, when the suitability petition approved, and then first ANDA coming in and submitted, and after the approval, then PSG was published. But now our new approach is, once the suitability petition is approved, we develop the PSG so that we give more clear guidance for ANDA development, so that and then we receive the ANDA, and then ANDA get approved. So this is like kind of flips, like now we have a guidance available from the start. And in my previous slides, in the pie chart, the most common petition type was the strengths. So now I'm gonna talk about the higher strengths, the PSG stress for strategies for petition higher strengths. So for study design considerations for in vivo, we consider and evaluate the formulation information and data on the food effects and strength waiver, and also BCS waiver, fat waiver, all those factors. And then for PSG resolution, we provide two options. So option one is basically when there's no approved product in the market yet. For this case, for higher strengths, applicants can use multi-units of the RLD compared to 1 unit of test based on dose proportionality PK and the formulation proportionality. And then option two, it's for when there's approved product in the market, and then that product designated as like RS. So it's like after first approved petition ANDA. For this case, it's similar to other B study, 1 unit of approved first petition ANDA, the RS, compared to the 1 unit of the test. And here's the example for higher strengths, which is the enzalutamide oral tablets. So before the suitability petition approval, the RLD, the tablet, there were 40 milligram and 80 milligram were available. And then the petition strengths, the higher strengths is 120 milligram and then 160 milligram. So there's no PSG for that higher strengths, and then there's no approved ANDA for this. And then we received control correspondence about the study design, asking our guidance on this for the higher petition strengths. So the PSG was revised to include the petition strengths. And here's the our two options in the PSG. So again, for option one, it's when there's no approved product yet for the higher strengths. So competitor would be the lower strengths, the 80 milligram of the RLD. Applicant use 2 of them to compare to 160 milligram test. And then the 120 milligram can be bioweight. And for option 2, when there's it's for after first petition and approved when there's RS. So similar to other B study, applicant can use the approved petition and the 160 milligram as a competitor, and bio waiver for the 120 milligram would be the same for option 1. So now we talked about the strengths. Now we're gonna talk about the dosage form, the new dosage form. So the factors we consider are also the same, like the formulation, food effects, but the thing is for new dosage form, we don't have such data. So we'll take more conservative approach for the options. So option 1 and 2, it's kind of similar strategy for strengths. Option 1, there's like no approved ANDA. So we ask compared to RLD per intended label use. And for option 2, it's for when there's approved product for that new dosage form. And I'll talk more when I share the case studies, which is memantine ODT. So the RLD is the memantine tablets, like the 5 milligram and 10 milligram. And then the petition new dosage form is the ODT. So PSG for RLD tablets, we have 2 options as species biowaiver, and then also in vivo PKB option because the memantine is considered as species 1. But there's no PSG for petition ODT. And then there's no approved ANDA for the petition dosage form. We received the control correspondence asking about B study design and then dosing instruction for ODT based on the approved petition. So we developed a new PSG to provide clear BE recommendations. So for option 1, there's no ODT available yet in the market. We recommend to use RLD tablets as the competitor. And then for B study design, we recommend 3-arm for memantine because for memantine petition labeling, approved petition labeling, it recommends to take ODT with versus without water, both are allowed. So for this case, for fasting study, we recommend 3-arm, like the RLD tablet with water, meaning they need to swallow the RLD tablet, so need water. And then we also recommend test ODT with water and also test ODT without water, the 3-arm, so that we have so that we ensure that all these 3-arm, because labeling says with or without water, have competitor exposure. And then we don't recommend fat for this case because it's species 1. Because most cases, if it's not species 1, we ensure to recommend fasting and fat so that we have data for the food effects. And for option 2, it's after the first petition ENDA. So meaning the ODT petition ENDA, there's a pre-petition ODT and then designated RS. The applicant can use that new ODT, the RS, as a competitor. And then for B study design for option 2, when there's ODT available, we recommend 2-arm study, RS ODT without water and test ODT without water as most conservative approach. And here are the challenges for suitability petition. Not much of data, but we ensure that we assess all the scientific considerations, but the impact is PSG have been proactively developed with the 2-option framework and science-based approach considering BCS, ICH, and 13A for especially for option 2 and also the dosage form. And here I'd like to express my sincere thanks to the PSG review team who was the initial pioneer, my team lead Zhang and Joy, Gang, Wen, Cheng, Ji-Hong, especially also to our director MJ, and then PSG team and also our ORS leadership for all the guidance and support for this suitability petition PSG. Thank you.
Thank you, Dr. Hailim, for covering this very important work and making sure that all applicants have access to these guidance recommendations to get alternative dosage forms approved to the market in a timely manner. Our next speaker is Dr. Yi-Hsien. She's a regulatory officer in the Division of Therapeutic Performance 2 in Office of Research and Standards at the FDA. And her talk today will focus on other general case studies for alternative bioequivalence approaches when the RLD is not available and there is no RS. So she will cover those specific cases from the regulatory lens. Give a warm welcome for Dr. Yi-Hsien.
Thank you very much, Heather, for your nice introduction. So good afternoon. Today my talk is about alternative bioequivalence approaches for generic immediate release oral solid dosage form drug product development when reference list drugs or reference standards are unavailable. Here is my disclaimer. This presentation reflects the review of my own opinion and the information I've collected. And sorry, it should not be construed to represent FDA's review or policies. Let's go through some basic concepts. We have talked a lot about reference list drug product ROD and reference standards RS. What are they? So the ROD is basically the list drug product that an applicant must demonstrate the sameness regarding the active ingredients, dosage forms, and route of administration, labeling, strengths, and other characteristics. So basically, ROD serve as the benchmarks for sameness. RS is the specific drug product selected by FDA that ANDA applicant has to use it for either in vitro and or in vitro either in vivo BE studies or in vitro studies. FDA generally select the highest strength of the ROD product line as the RS based on without any safety concerns. When ROD is not available, FDA generally selects and approve the ANDA served as RS. They have to meet the market leader, also containing the complete strengths of the entire ROD product line. Last, as
Helen just mentioned, FDA will designate the first ANDA approved through the suitability petition as RS in different dosage forms or different strengths, which is not included in the ROD product line. So why does this matter? So discontinued ROD RS of IR solid dosage form drug products, they created significant barriers and challenges for generic development, and due to lack of the appropriate comparator to demonstrate BE. And also generic applicants are continuing to express their interest in developing those discontinued drug products by submitting the control correspondences and ANDA meeting request the agency's advice on potential alternative B approaches. So the solution to overcome this regulatory barrier, we try to identify the viable alternative B approaches to guide the generic drug development even when ROD, RS are discontinued. That's what we have done. We have collected the data on discontinued ROD product from Orange Book. Then we surveyed our prior recommended B approaches from either responses to the control or pre-ANDA meeting request. Eventually, we established the framework of alternative B approach to demonstrate BE even when ROD are discontinued. Here is our regulatory navigation of discontinued ROD and RS. So if the ROD is discontinued due to safety and effectiveness reason, then this ROD will be permanently removed from Orange Book. Therefore, it's not 505 jailable. No ANDA should be developed. On the other hand, if the discontinuation of ROD not due to the safety and the effectiveness reason, and then we have classified them into 3 groups. Subcategory 1, no ROD, but we do have approved ANDA generic drugs on the market, so which can be designated as RS. Category 2, no ROD, neither for RS. However, we do have approved product with the same API in other dosage forms on the market. Subcategory 3, neither ROD, RS available, and none of any other product with the same API on the market. So basically, you can see for subcategory 1, we don't face any regulatory challenge. However, for both subcategories 2 and 3, we do face regulatory challenges, and there is a regulatory barrier for us to deal with. We have done the survey of discontinued ROD for IR solid dosage form per our Orange Book. Here, this pie chart shows the landscape of the distribution of each subcategory based on the total of 186 discontinued IR solid dosage form products. 54% for subcategory 1, 21% for subcategory 2, and 25% for subcategory 3. As we all aware, without the generic development, due to the ROD discontinuation, it does cause public health impact. You can see from this stacked bar chart, multiple therapeutic areas are affected across the board, especially for oncology drug and cardiovascular and so on and so forth. Here is a framework for alternative B approaches we have developed. As I mentioned earlier, the challenge majorly focused on subcategory 2 and 3. For subcategory 2, because we do have other dosage form approved product on the market, so the alternative B approach will be using cross dosage form bridging method to demonstrate BE. For subcategories category 3, as there are no available same API drug product in any dosage form available, so the alternative B approach will be through the relative bioavailability studies. So the test of product can compare with the appropriately formulated oral solution. And in addition, the BCS3, 1 and 3 based biowaiver definitely will eliminate the in vivo study if the drug meeting the solubility, permeability, dissolution, especially for 3 and formulation Q1, Q2 similarity requirement. I'm going to quickly go through a couple examples by using this established framework for alternative B for those discontinued ROB. Example 1, aloxipalanosatrum hydrochloride oral capsule, which is indicated for prevention the chemotherapy-induced nausea and vomiting. It only have one single strength, 0.5 milligram, and which is not discontinued due to SE reason. So the alternative B approach is through the BCS class 1 based biowaiver because it's highly soluble and highly permeable. Example 2 is oncology drug, Gleevec oral capsule, which is containing both strengths, 50 milligram and 100 milligram. Both strengths were discontinued not due to SE reason. So the alternative approach, we recommend compare the imatinib mesylate oral capsule to the alternative comparator, Gleevec oral tablet, to demonstrate the VE. Example 3 is a Dartisla ODT oral disintegration tablets, which is indicating for treatment of peptic ulcer, and it does have 2 strengths in its product line, and both are discontinued. The alternative B approach we recommend to compare glycopyrrolate ODT to the alternative comparator, Rabinol 40 glycopyrrolate oral tablet to demonstrate VE. Example 4 is an anti-infectious drug product, Cytovan ganciclovir oral capsule. It has both 250 milligram and 500 milligram strengths. Both are discontinued, not SE reason either. So the alternative B approach, we recommend compare ganciclovir oral capsule to the alternative comparator, ganciclovir injectable, to through this absolute bioavailability comparison to demonstrate VE. The last example is reserpine, which is an anti-hypertension drug. And it has 0.1 milligram and 0.25 milligram strengths in this product line, which are both discontinued. We recommend to compare the reserpine oral tablet to the alternative comparator, the appropriately formulated reserpine oral solution, because this drug belongs to subcategory 3. There's no approved product containing same API in any dosage form on the market. So in addition to the alternative B approach I already showed in our framework, this is our future horizon. We already heard many speakers talk about the quantitative medicine and the virtual bioequivalence. What they can offer us, they can use the physiological data to predict the in vivo performance of the drug product. and then establish the virtual BE standard. And then the other benefit is to replace the in vivo bio study and also provide a solution to discontinue the ROD. In summary, so this project has provided innovative scientific and regulatory impact, address a regulatory challenge for generic industry due to discontinue the ROD-RS, establish the strategic framework for alternative B approaches can be applied to each scenario, provided the scientifically sound pathways to demonstrate BE and mitigate regulatory barriers for generic drug development, eventually promoting the public health by preventing drug shortages, maintaining therapeutic options, and ensure access to the critical medicines and support safe, effective, affordable generic medicines. So I want to conclude my presentation with the following key messages. First, the proposed framework provides practical solutions to the critical regulatory challenges due to discontinued ROD-RS. And second, with the established framework of alternative B approaches due to the discontinued ROD-RS, we strongly encourage the generic applicant engage with the agency at the early stage, either through the controls correspondences or pre-end meeting requests to discuss the viable alternative B approach. And this is, I have to thanks all the members in our research team and also the management of my division and OSIO. Thank you.
Thank you, Yi-Hsien, for walking us through those different categories of creative solutions for how to go about approving a generic product when no reference is available. So our next few talks will be more problem-solving specific for this topic. So our first speaker in this part of the session will be Dr. Brooke Langevin, who is an assistant professor at the University of Maryland. And she will talk about her vision for use of innovator studies and virtual reference data to support the BE bioequivalence assessment of generic products when there is no reference listed drug available. So welcome, Dr. Brooke Langevin.
Thank you.
Hi, thank you very much for the kind introduction. So today, as was mentioned, I'm talking about a little bit of a shift from what's been discussed and looking at a potential framework for assessing BE when the RLD or RS is unavailable. As you heard earlier in the session, there are a not insignificant number of products where there is an interest in developing a generic product. However, the RLD or RS are not available. In the presentation just prior, we saw a potential future solution in terms of virtual bioequivalence trials, making use of prior information. So when there are no specific RLD or RS products available, we still have fragmented evidence sources. We have information from historical studies. Nowadays, there's often a literature report containing PK studies that have been published, including a PK model. And there can be other external data from the literature, both from the review and just papers published outside of that. And so the question really becomes, is there a way to make use of all of this data and try to have a systematic framework for evaluating which types of methods in which scenarios make sense in terms of establishing BE? So one of the questions and the big question posed is, how do we ensure decisions remain consistent and defensible, but are still supportive of the timely generic access? So some of the potential solutions are to look at the alternative reference standards, which we've talked about earlier this morning. And the second is to see if there are, another potential solution is to see if there's a way to use these available data from these innovator legacy studies from the literature to try to create a hybrid approach that can still assess BE. One of the issues with this that is often presented is that they rely on assumptions about the reference data that you're pulling from that are not always directly observable and can influence the bias, type 1 error, power, and all requiring explicit evaluation when BE inferences will remain reliable in the absence of an RLD and when they can lead to the wrong conclusions. So the potential solution is a way to evaluate the different methods for establishing BE with the use of a virtual reference bank. So the overall framework is to first create and I'll go step by step through each, but it's to first create this reference bank and then to characterize the data that you have both in the test study and the reference bank construction, and then to evaluate different methods for BE based off of that characterization, and then to conduct the assessment. So the first step, and arguably the most important, is actually constructing this reference bank. So I mentioned that oftentimes there's literature or PK model data that can be used to simulate and generate a virtual reference population for a reference bank. However, that is not necessarily, we can't just take those profiles and immediately go and conduct a BE assessment with those profiles. There are factors that have to be always accounted for example, the variability within the reference bank data, some of the covariates, especially if there are significant covariate effects when we're talking about comparing it to a test study, trying to make sure that there is reasonable overlap between the test data and the reference data that is constructed. And in the absence of this reference data, or in the absence of these PK studies, is there a way to make use of other information from the innovator studies? A lot of that would require collaboration between the FDA and industry support to try to create both a PK model that could be then established through a framework like the model master files when there is significant need for a generic, but those PK models don't already exist in literature. Something like a model master file could be used to create this virtual reference bank and released in that method. When we're characterizing the data, these are the factors that we've identified as potential factors for which type of BE assessment will maintain the operational characteristics that we're expecting, and that will allow for BE assessment. So the first within the reference bank is talking about the variability that is established in the reference bank, the overlap of covariates between the reference bank and the test study, the sampling density in the test study, the distributional assumptions made both from the actual test data itself and the distributional assumptions from the reference data, and any model misspecification that was established in the creation of the reference bank. So each of these factors in combination will affect which approach may be the best approach, and if any of these approaches are actually feasible for BE assessment. So we've kind of broken the approaches that we're considering here into two categories, a statistical approach and a model-based approach. So the statistical approach is looking at simple resampling from the reference bank in terms of your key NCA parameters, such as ACC max, Monte Carlo approaches. And then for the model-based, This is your more traditional MIBE approaches, where we're looking at the reference PK model and then re-estimating for the test data and using simulation-based inferences. And so within each of these categories, those factors from the data characterization are going to impact the overall results. So for example, when we're looking at factors like an unusual distribution in the reference data, the statistical approaches often fail to preserve type 1 error and power. And they show significant bias, whereas the model-based approaches are expected to perform better. And so the next step in this framework is to look at the actual BE assessment. So in the proposed framework, you would look at actually performing simulations with a known ground truth that matches the operational characteristics of your reference bank and your test data to actually create a simulation to assess some of these operational characteristics that match your data characterization to see to confirm that power type 1 and bias would be expected to be preserved in the use case that you're considering. So for any drug product, you would first have to make sure that that a ground truth simulation would help support that these characteristics would be maintained and that you would not have difficulties, that you would not show signs of these operational characteristics being different, because these simulations have to be conducted through the full range of different operational characteristics, especially when you start talking about more complex dosage forms or more complex profiles. So the overall goal would be that this framework could be tested across a wide range of operational characteristics and data characterization to allow for a general framework that could be easily established for a variety of products where it wouldn't have to be tested individually each time, and these reference banks could actually be used across a wide variety of products, not necessarily something that had to be company specific. So for a given reference product, this reference bank could be constructed and be used for several generic development goals and characterization, several development projects, and be used to actually help support several products that were being developed. And BE could be included. The BE conclusion could be established based across these characteristics. Obviously, one limitation to mention is that each product would have to account for the data context and the assumptions made. So any of the assumptions made in the data characterization phase would have to be pre-established in order to be sufficient. And overall, this approach can only be considered within the totality of evidence and making sure that all of the previous steps aligned and supported the overall conclusions. So the overall strength of an approach where we have a framework to characterize several, that would allow for the characterization of several different products and be able to state which approach would work for a given data characteristics is that it allows for these methods to be tested and stress tested under controlled conditions, and allows for a structured and reproducible framework that once used several times would allow for a much faster flow when in situations where all the RLD and RS are unavailable. And it would be able to be operationalized. So it's something that we envision in order to really support access could be operationalized within AI to allow for these checks to be run independently and to help support faster development. So you could match your data characteristics that you observed and the different limitations and which method to be a much faster runtime. The limitations are this is always going to depend on the quality of the available data. So when the literature model isn't good and the data isn't there, it's not a feasible approach. And it's going to be limited to the scenarios that have been pre-evaluated. So like I mentioned, those data characteristics have to align with the pre-tested scenarios, and it hasn't been yet applied. So I'd just like to say thank you to everyone at the University of Maryland, and thank you for giving me the opportunity to present.
Thank you, Dr. Langevin, for sharing your thoughts on this topic. Our next talk that we have in this session is going to highlight our work on developing the first product-specific guidance that offers an alternative bridging approach for a scenario where there is no RS. And this work was a tremendous effort across the agency and includes a lot of different characters whom will be acknowledged. But today we have Drs. Robert Hopefl and Dr. Wanjie who will give us the rundown on that story. So welcome.
Okay.
All right. Good afternoon, everyone. My name is Robert Hopefl, and I work in the Division of Quantitative Methods and Modeling. And this will be a co-presentation with my colleague, Dr. Wanjie from the Office of Biostatistics, and she'll be covering the second half of this presentation. So our presentation today is on the PK bridging approach to RLDs via alternative comparators. Okay, so to give an outline of our talk today, I'll give some background about the general BE approach using PK endpoints, and then also introduce the general bridging approach to establish BE to the discontinued RLD. Then we'll go over 2 case examples. And then also in the second half of the presentation, my colleague will present the more statistical considerations for applying this approach. Okay, so I believe we kind of sufficiently covered this information already today. So in the interest of time, I'll just move on to my next slide. So our talk today will focus on the 2 recently published PSGs that included this option to perform an alternative BE approach using an alternative comparator to bridge BE to the discontinued RLD when there's no reference standard available. So these PSGs are for trazodone hydrochloride ER tablets, published in October 2025, and then for potassium chloride ER oral suspension, published in December 2025. And in both of these PSGs, this alternative approach is placed under a second option in the PSG page. So this slide gives an overview of the general bridging approach. So in these cases, you can see that the RLD is discontinued, but there is some type of historical bioavailability data that compares the discontinued RLD to the alternative comparator. And this alternative comparator is something that's currently on the market. And then what a generic applicant would do is do a new PK study comparing the proposed test product to the alternative comparator. And then using the information from both of these 2 separate studies, the final BE decision can be determined, and it depends on whether the confidence interval is between 80 to 125. And throughout this presentation, we refer to the alternative comparator in the historical study or study 1 as C1, And then C2 denotes the alternative comparator in the new PK study or study 2. So this approach, again, provides generic applicants a pathway that are interested in developing products when there's no RLD or RS available on the market. And this was developed in heavy collaboration with the Office of Biostatistics. And specifically for these cases, we cannot apply BE transitivity because this discontinued RLD is not bioequivalent to this alternative comparator that is on the market. So this brings us to our first case for the trazodone hydrochloride ER tablet. So this slide summarizes the historical data. So the RLD was approved as a 505(b)(2), and the NDA holder did a relative bioavailability study comparing its ER tablet to an immediate release tablet. And this was done in a two-way crossover study and comparing a single dose of the ER tablet to 3 doses of the IR tablet given throughout the day. And ultimately what they found is that given the same total daily dosage, that the ER tablet given once is not bioequivalent to the IR tablet given 3 times throughout the day. And you can see a figure of the PK for the ER tablet and IR tablet, and then also the statistical summary results in that table there. So this brings us to our recommendations in the PSG page. So this is for what we call study 2, or what the generic applicant would perform. So we have one in vivo fasting VE study with PK endpoints. And the alternative comparator here, or C2, is the trazodone hydrochloride IR tablets given as 3 times 50 milligrams, and that's given throughout the day. And that's compared to the proposed test product, which is a trazodone ER tablet, and that's 150 milligrams given once. So this recommendation of a total dosage of 150 milligrams in the PSG is due to the increased risk of QT prolongation when administering a 300 milligram dose of trazodone in healthy subjects. And we're able to make this adjustment from 300 milligrams in the historical study to 150 in the new PK study because the ER and IR tablets are dose proportional in terms of PK over this dose range. And also, we don't recommend a Fed BE study in this case due to the increased risk of QT prolongation under Fed conditions with trazodone. So this slide kind of gives an overview of how this approach would be applied. So the figures on the left represent the historical data and study, and then the figures on the right would be our hypothetical results that an applicant would have after doing their new in vivo PK study. So here you can see that this approach uses the statistical information from both the historical study and the new in vivo PK study to ultimately derive the final TR ratio and 90% confidence interval. And again, my colleague from biostatistics will go over more information on that in the second half of the presentation. But this approach overall uses the relative PK difference based on each study. So for example, the highest peak of the IR formulation in the historical study would be compared to the highest peak of the ER formulation in the historical study. And then that difference would be compared to the highest peak of the IR tablet in the new study versus the highest peak of the ER tablet in the new PK study. So this brings us to our second case example for potassium chloride oral suspension. And in terms of the historical study, the NDA holder did a relative BA study comparing the 40 milliequivalents of the ER suspension to 40 milliequivalents of an ER capsule. And this capsule is also an RLD. They also did a baseline correction because it's an endogenous compound by subtracting the control period from the dosing period. And ultimately what they found is that the RLD ER suspension was not bioequivalent to this RLD ER capsule. And another complexity in this case is that the RLD ER capsule is also discontinued. but we have an available generic RS that's on the market that references this RLD capsule. And then similarly, you can see the PK in terms of rate of potassium excretion for the RLD capsule and RLD suspension, and the PK results in the table on the bottom. So in terms of our recommendations in the PSG page, we recommend one in vivo fasting VE study with PK endpoints using the alternative comparator of the RS potassium chloride ER capsule. And this is in a total dose of 40 milliequivalents, and that's compared to the proposed test product of the potassium chloride ER suspension in a total dose of 40 milliequivalents. And this 40 milliequivalent dosage is recommended due to the availability of reconstitution methods that come from the RLD studies. And also for this specific case, we do not recommend Fed-BE studies due to precise potassium level control that's required for potassium BE assessment. And another complexity for this specific case was that the historical data was not previously publicly available. So in this case, we're able to include the necessary statistical information to use this approach within the PSG. And also we included some in vitro dissolution testing using the test product because the RLD labeling states that it can be administered after sprinkling on applesauce or reconstituted juice. And we're not able to do a similar PK bridging approach because there's no NDA sprinkle study available in the NDA to complete the bridging approach. So this is just a summary slide that I have prepared, but what I want to call out here is that if applicants are interested in applying this PK bridging approach or other alternative BE approaches, that they're encouraged to come and discuss with the agency. And I have some links to different meeting pathways, as well as the guidance to help determine whether to submit an ANDA or a 505(b)(2). And with that, I'll pass it to my colleague, Dr. Sun, to cover the statistical considerations.
Thanks, Robert. Good afternoon. My name is Wanjie Sun. I am from the Office of Biostatistics, DB8, and I'm going to discuss the statistical considerations in using alternative comparators to establish bioequivalence. So as several speakers already mentioned this, so we have used the RS in establishing bioequivalence. And this RS actually is an alternative comparator. So when the RLD is discontinued for non-safety nor efficacy reasons, if we already have a generic drug on the market and we may designate this generic drug just as a RS, and then in the new ANDA study, we can just compare the test product to this RS. And the question of interest is this test product bioequivalent to the RS or not? If the answer is yes, then we claim that the test product is bioequivalent to the RLD. So the ultimate question is whether the test product is bioequivalent to the RLD or not. However, we do not really bridge back to the discontinued RLD, and there is scientific justification behind this, which I will discuss later. Similarly, we also use a bioequivalent alternative comparator to establish bioequivalence. Again, if RLD is discontinued for non-safety nor efficacy reasons, and if we have an alternative dosage form that was approved to be bioequivalent in a historical NDA study, then we could use that alternative dosage form of the same API as an bioequivalent alternative comparator. In the new ANDA study, the test product can just be compared to this alternative comparator. And then again, if the bioequivalence assessment test passes, then we say that this test product is bioequivalent to the discontinued RLD. Again, we do not bridge back to the original RLD. So in both cases, there are actually two underlying assumptions here. The first assumption is the constancy assumption by Lee 2007. What this means is that the RLD has the same bioavailability between the historical and the new studies. For example, the study design is the same, inclusion-exclusion criteria is the same, study conduct is the same, methods of determination for plasma concentration is the same. Of course, in practice, like you cannot have two exact the same studies. Also, over time, study practices evolve. However, the new study should be designed as closely as possible to the historical study when feasible. And then the second assumption is bioequivalence transitivity by Anderson and Hogg 1996. What this means is that if A is bioequivalent to B and B is bioequivalent to C, then we conclude that A is bioequivalent to C. So as you heard from the first speaker of this section mentioned that there were about the bioequivalence shift. So I will discuss this on the next slide. So in practice, as we said, data may deviate from these two assumptions. However, these two assumptions provide a scientific justification for the current regulatory practice. So when we use bioequivalence transitivity, so there are advantages there. So for example, it's easy to implement for the sponsors, and we can reduce variability, and the confidence interval is narrower than if we use the bridging approach, which I will introduce next. Of course, there is no free lunch, right? So one potential caveat is that, how about the shift, bioequivalence shift? So is the bioequivalence transitivity assumption met or not? Well, there are different factors you need to consider, which I will not elaborate on here. But good news is that in the majority of the cases, the answer is yes. David et al. And our FDA colleagues back in 1997 analyzed over 2,000 ANDA studies that were approved between 1996 and 2007. They found that the average difference in Cmax and AUC between the generic drug and the innovator products was 4.35% and 3.56%. So what does this mean? This means that the GMR, the average GMR is about between 95% and 105%. And then also in nearly 98% of the bioequivalence studies, the generic drug product AUC differed from that of the innovative product by less than 10%. So for 90% of the studies, the GMR is bounded between 0.9 and 1.11. So generic drugs actually are very close to the innovator products. This helps to meet the bioequivalence transitivity assumption. Now, the challenge is that what if the alternative comparator is not bioequivalent to the RLD? Then we cannot really apply this bioequivalence transitivity, and this leads to the statistical bridging method. I will use the trazodone case to discuss the statistical considerations for bridging approaches. So in this one, as Robert already introduced, so in the historical, the RLD of ER tablet 300 milligram, this one is out of market for non-safety nor efficacy reasons. However, in the historical NDA study, and there is no RS product, generic RS product. But in the historical NDA study, we do have the IR tablet 300 milligram in the same relative bioavailability study. And for this one, we call this C1. And we can use the, however, okay, these two formulations, they are not bioequivalent. However, we can still use this IR tablet as a bio-inequivalent alternative comparator. So in the new AND study, we could compare the generic ER tablet to this IR tablet. As Robert introduced, we're recommending 150 milligram due to safety efficacy ethical considerations. So this new, the IR tablet in the new AND study, we call this C2. Here you can see that the question of interest and the ultimate question is the same, different from before. So our question is that, is this test product bioequivalent to the original discontinued reference RLD or not? So in the PSG, you can see that the study design is two-way crossover design. Formulations, as we said, we have the ER tablet 150 milligram versus IR tablet 150 milligram used as the alternative comparator. So we have two assumptions here, a constancy as I introduced before, and also we need the dose proportionality for both ER and IR products. This is to bridge different strengths across the two studies. And for the proposed statistical methods here, so we need the point estimate and also the confidence interval for the GMR. For the point estimate, the log transformed GMR for T versus R is the summation of the log of GMR between the two studies. Intuitively, we could see that this one, T over R equals T over C times C over R. Under the assumptions of constancy and dose proportionality, we can cross out in this to C. So this is how this bridging works. Intuitively, it's like just like this. And then the 90% confidence interval can be derived as follows. First, we need to test whether the inter-subject variance is homogeneous between the two studies by the F-test. If the inter-subject variance is homogeneous, then we can calculate the 9% confidence interval by using the recommended formula in the PSG with the pooled variance. And if the inter-subject variability is heterogeneous between the two studies, then we could use Welch-Satter's weight approximation and to calculate the 90% confidence interval. You can just follow the PSG. The formula is provided there. We conducted hypothetical case studies over a range of plausible scenarios, and we verified that the bioequivalence between T and R can be achieved by reasonable sample sizes with adequate power. And this is the PSG with the formula. So the bridging methods published in the PSG for trazodone, this can be used as the general bridging method, which can be applied to other products. However, the detailed bridging methods may require case-specific modifications depending upon a holistic considerations of regulatory history, sponsors' data access limitation, proposed bridging pathways, study design of involved studies, dosage forms, strengths, PK profiles, bias and efficiencies of the estimated bioequivalence effects, sample size, and other considerations. Again, applicants, you're recommended to discuss your development plan with the FDA where pre-end meeting requests. So some take-home messages here. This innovative statistical bridging approach may provide a scientifically sound and practical alternative bioequivalence approach for generic applicants to establish PK bioequivalence when there is no RLD, no generic RS, or bioequivalent alternative comparator available on the market. It helps to facilitate the generic drug development and improves patient access to affordable generics when there are challenges to establish bioequivalence with existing RS. Again, applicants should discuss your approaches with FDA to ensure that your proposed generic drug is appropriate for submission as an ANDA. With that, I'd like to acknowledge my colleagues from OGD and Office of Biostatistics. This is a wonderful collaboration across multiple disciplines. With that, thank you for your attention.
Thank you, Wanjie-san. Moving on, our last talk for the session will be by Dr. Pamela Dorsey. She's a senior pharmacologist in the Office of Bioequivalence, and she's going to offer the final perspective from the bioequivalence reviewer perspective on this topic and provide us some unique case studies from that perspective before we head into the panel question and answer session. So welcome, Pamela.
Thank you, Heather, for that kind introduction. Let me play with the clicker here. Okay, there we go. So my talk is kind of a piggyback on Yi's presentation earlier about different pathways that applicants can use for determining BE for their drug product. In the event that the RS is unavailable, my talk, I want to highlight control correspondences that we received where we were actually able to give applicants the ability to use a different dosage form as a comparator product. So this is my disclaimer slide. Of course, these are my views and should not be construed to represent FDA's reviews or policies. So the background for my talk, I very briefly will provide the definition of a RLD and reference standard. However, I feel like we've drilled that into the ground already. Next, I'll go into control correspondences that we receive from applicants asking for an RS designation due to some issue that they come across. Then I'll go into possible alternate bioequivalence approaches that we provided to applicants to kind of get them out of this quagmire with what to use. And then I'll segue into transitive bridging. And this is kind of a topic over using a different dosage form and how we're able to do that. And I believe Wanjie just provided a very good summary on that as well. And the main meat of my talk today is focusing on 3 case studies that we received as control correspondences within the Office of Bioequivalence, asking to use a different dosage form as an alternate comparator product. I want to end with kind of guiding principles for an alternate B approach. If you're looking to use this approach in the future, things that we really recommend or strongly encourage that you provide within your control correspondence to the agency, and of course, a summary. So we have already gone over this. So the RLD, just your basis of submission for your ANDA, and of course the RS is just the drug product that the FDA has selected for you to conduct your in vivo bioequivalence studies. As I mentioned prior, we received control correspondences within the Office of Bioequivalence from different applicants asking for us to designate a reference standard or provide an alternate comparator product. And I believe this was a great summary from the first talk of this session. There can be a number of reasons, some being that, you know, the electronic Orange Book, they have no RS listed, or the current RS is discontinued. Again, limited distribution, or it hasn't been marketed at this point. Or the applicant believes that another approved product may be more appropriate to use. So some solutions that we have been able to provide applicants in the past for some of these control correspondences, of course, to possibly utilize and authorize generic of the RLD, of course, this is the most straightforward. To utilize a different strength of the RS. So if the RS is 100 milligrams, it's you know, no longer on the market, you can't procure it. Possibly using 2 times 50 if 50 is part of the product line. Utilizing individual components for fixed dose combination products. And in Robert's talk, possible PK bridging to the RLD when B is not directly established with an alternate comparator. And again, for my talk, I want to highlight what happens when you are able to use a different dosage form for the same drug product with the same route of administration. So this is a very basic slide on transitive bridging. And again, we touched on this prior, but you have that historical BE study. It can be BE or BA, where the innovator compared the reference standard 1 with dosage form A to reference standard 2 with dosage form B. If BE was determined between those 2 dosage forms, and we have enough PK information to have a scientifically sound conclusion, then the applicant can come forward and conduct that the B study 2, where their test product with dosage form A can be compared to that second reference standard with dosage form B. If you determine BE within that study, then we have the kind of basic conclusion of your intended test product with dosage form A would be BE to the reference 1 of dosage form A. So again, I said prior that the case studies that I'm going to provide are control correspondences that we received within the office asking us, hey, I'm stuck. Is there a way that I can use an alternate dosage form for the same drug product in order to get my study done? So the first case study, and I'm not allowed to disclose this information for this one, but this is an oral suspension. Of course, the RLD is discontinued, and there's no RS that has been designated for this product, no approved ANDAs. The applicant came forward to us to ask if it was acceptable to conduct BE studies comparing the suspension to the immediate release tablets. So within the body or within the letter of the control correspondence, the innovator actually provided the label that contained not only the suspension itself, but the immediate release tablet. So the dosing regimen was the same for both. They also provided published literature demonstrating BE between the oral suspension and the immediate release tablets, as well as the product-specific guidance for the immediate release tablets as a basis for the proposed study design. So how are the suspension and the immediate release tablets related? How are they BE? So within that published study, they provided, it had a study where 2 lots of the immediate release tablets were compared to the oral suspension in a 3-way crossover study design under fasting conditions. And of course, the suspension was determined to be BE to the immediate release tablets. So based off of the information that we received from the applicant's approach for conducting two-way crossover studies under fasting and fed conditions, comparing the suspension to the immediate release tablet, we deemed acceptable based off of the same dosing regimen for the oral suspension and the immediate release tablets per the innovator label. And also that publicly available published literature that the applicant was able to provide to us demonstrating BE between the suspension and the immediate release tablets. So following the response to this CC, the applicant actually filed an ANDA for the suspension, comparing their suspension to the immediate release tablets, and they were indeed able to demonstrate BE between their suspension and the tablets under fasting and fed conditions. For case study 2, I can disclose this one. This is for buspirone capsules 5, 7.5, 10, and 15. The RLD is discontinued. And at that time, there wasn't a reference standard for this particular product. So this potential applicant came forward asking for concurrence to utilize buspirone tablets 15 milligram as a comparator product to demonstrate BE for their capsules. So won't go into details with this slide, but just kind of an overview of the relationship between the capsules and the tablets. For approval of the capsules, they demonstrated BE to the innovator for the tablets using the 15 milligram strength. Before the innovator for the tablets, before they were discontinued, they actually demonstrated BE to ANDA 075, 022. And that once the innovator of the tablets became discontinued, and a 075, 022 became the RS. And for the current RS and a 202,557 for the buspirone tablets, BE was demonstrated between those two using the 15 milligram strength as well. So again, based off of the information that we had received, the applicant's approach to conduct two-way crossover BE studies under fasting and fed conditions, comparing the capsules to the tablets, was deemed acceptable. This was due to publicly available data that they had been able to provide us within their control correspondence, demonstrating BE between Buspar tablets and Buspar capsules. And this was actually information that was at drugs at FDA, the redacted reviews within drugs at FDA. And they also provided the PSG for Buspar tablets as a basis for the proposed study design. And using this approach, after we had sent the response back, we actually have a currently approved ANDA using this approach. For case study 3, this is an orally disintegrating tablet, 10 and 20 milligrams. Again, the RLD is discontinued, no RS due to no approved ANDAs. So this applicant came forward to the agency to ask if they could compare the immediate release tablets as using the immediate release tablets as a comparator product to demonstrate BE for their orally disintegrating tablets. So this is simply a slide on how the relationship between the orally disintegrating tablets and the immediate release tablets. So there are 2 studies for approval. The first being a single dose, fully replicated study comparing the orally disintegrating tablets to the immediate release tablets under fasting conditions. And this was given, water was given for both with this one. And BE was demonstrated between both dosage forms. The second study that was conducted was a 2-way crossover study comparing the ODT administered without water to the immediate release tablet administered with water. And again, under fasting conditions, BE was demonstrated for both dosage forms. So for this applicant's proposal to use the immediate release tablets as an alternate comparator for demonstration of BE for the ODT was deemed acceptable. And I want to highlight again, this was within the letter for the control correspondence. This was information that the applicant took from redacted reviews within drugs at FDA. So I want to pause right here and maybe I'll start with the second bullet. When you are submitting a controlled correspondence and you feel like you can possibly use an alternate BE approach within your controlled correspondence, please state that fact that you are seeking to use an alternate BE approach, and do this early within product development. And in doing so, if you have any publicly available data, published articles, redacted reviews, anything that you can provide as justification for your approach, we strongly recommend that you submit that with your control correspondence to the agency, as well as any viable study design that you may possibly consider to determine BE for your intended drug product. And I just want to highlight any publicly available information that you have, please, please, please submit it with your controlled correspondence. So I'm fast. This is my summary. When an RLD-RS is not available and there are no approved ANDAs, demonstration of BE using an alternate comparator product of a different dosage form may be achieved through transitive bridging by utilizing publicly available data. And I say may, but this is definitely a pathway that could be taken to allow you to demonstrate BE between the drug product of the original dosage form and the comparator product. And always in summary, exploring alternate BE approaches allow for continued patient access to therapeutically equivalent drug products while maintaining the integrity of the ANDA approval process. And with that, I would like to thank the following people from OBIO, my division leadership, people that actually worked with me on these controls and the ANDAs themselves, and our ORS colleagues. Thank you so much.
Okay, we're almost at the home stretch. I'm going to invite all our speakers back up to the stage for the panel session. I also would like to welcome Dr. MJ Kim, who is the director of the Division of Therapeutic Performance 2 in the Office of Research and Standards at the FDA, and also Dr. Yuqing, who is a pharmacologist in the Division of Quantitative Methods and Modeling in the Office of Research and Standards at the FDA. Welcome. We will be prioritizing questions in the live audience, and Dr. Sivacharan will also screen for questions online. So please submit your questions if you haven't already. We will end the session on time at 4:05. I know there was a lot of information covered. I know there's probably a lot of really good questions out there, so let's hear them. And I already see somebody lined up. Go ahead. Yes.
Good afternoon. If the panel allows, I have two questions. First question is, in limited available RLDs, if a biowaiver is possible for lower strengths, can we use a combination of different strengths of RLDs? Because the reference standard strength is not available at the required quantities, and do a bioequivalence based on the dose normalized pharmacokinetics. As an example, if 75 milligram is the reference strength, there is a waiver possible for 50 and 25. In a clinical trial, can we use different strengths of RLD and present bioequivalence based on the dose normalized pharmacokinetics? That's the question number one.
Perhaps Dr. Yuqing Gong and Dr. Robert Hopefl might have some opinion based on the trazodone example.
I just want to clarify your question. I think you are asking, can we use different strengths to perform the BE study if the R&D strength is not?
Want me to repeat the question? I'm sorry. Just an example, these oncology medicines, they are very limited quantity, and we cannot get a sufficient quantities of a, let's say 75 milligram strength oncology product. And the manufacturer releases only certain quantities and they are not available for BE studies. In that case, can we use portion of the patients with a reference strength and remaining with a different lower strengths because the bioequivalence is possible for this on dose proportionality?
I think maybe Pamela and Yi want to address this. I think you're asking mixing the strengths.
Yes, portion of it. Let's say we have 100 patients in the study. Can we dose 50 patients with available 75 reference strength and remaining with whatever the available lower strengths and do a dose normalization and establish bioequivalence? That's a hypothetical question, but I want to ask.
I think hypothetically, it might be possible if you have dose proportionality and everything, but I do see the challenge here when you are doing patient study, when you have different strengths there, you may have more thinking to consider when you dose normalize. Like for example, do you have the same for dose normalization? Can you apply to both test and reference product? Are you assuming the test product also follow the same dose proportionality as sort of reference products?
Right.
And are you having the balanced test and reference in your trial? Are you dosing the balanced population with your higher strengths, lower strengths? I think it's a very complicated question. It's not a simple yes or no. Yeah.
Okay. Second question is, can we test two test products against a reference one RLD and submit as two applications, one as a 505(b)(2), one as a generic? Because the critical thing is lack of RLD availability. We have sufficient RLD, the sponsors do two studies in India. So here, instead of two reference products, we do two test products against one, 3 arms crossover or whatever, this thing. And it has two applications. Is that allowed or it is, again, a hypothetical question?
This is MJ, Kim. And I really appreciate your very challenging and complex questions. But I do also want to be mindful about some of the aspects that we are not hearing from your question. So in terms of the specific type of question, I believe it's a little bit difficult for us to give response. And we are more than welcome to talk with you after this panel discussions is over, or even a small group discussions. But you have heard from our presentation throughout, from these wonderful speakers, how the agency is trying to be innovative and proactively trying to utilize all available means and mechanisms to have these products available for the patients, right? So I hope you are not just not just limited to your end, but I hope those people who are calling into this webinar, you can hear from our end that we have shared really, really wonderful examples. And as I was sitting here listening to all these cases and these innovative approaches, I mean, I have to give kudos to the agency as well as the other speakers who have given this type of different things for us to consider. But we are more than welcome to talk with you on your specific aspect.
Okay, thank you.
Thank you so much.
Okay.
Hi, this is Fang from Office by Equivalence FDA. I just have a question regarding the alternate approach when the alternate comparator is not bioequivalent to the RD or IS. So now we give this bridging statistical approach, which is very good, scientifically justified. My question is just like from the pharmaceutical development perspective. So if I have a product is bioequivalent, I want to design, I have the comparator, I want to design a test product as same as the comparator. But now the challenge is I have to design a product which is purposely different from the comparator based on what our new statistical approach. Just I want to, maybe our the industry friends can shed some light on how challenged it will be, because especially for the ER product. Today we have we discussed 2 ER formulation, 2 ER products. And so because if you have the comparative bioequivalence, so you can use the dissolution, you can investigate the formulation design, and you want the dissolution as close as possible. But now since I have a different comparator and I have to purposely design a different product, with maybe, and sometimes maybe AUC has to be very close and Cmax maybe have to be higher than the comparator. Just like to be from the realistic perspective, how challenge it will be from how to develop such product. That's my question. Thank you.
Thank you. So from what I understood the question, if it is relevant to the trazodone example, which has been discussed, from the industry perspective, I think even though your comparator is changing, ultimately what we are talking here is that you're trying to build the bridge between what is available in the literature to begin with, like the actual RLD product has established its equivalence against that comparator. And now you are using that comparator to establish an equivalence. So definitely in terms of the CMC, there will be challenges because you don't have a actual comparator to test in vitro evaluation. So you are kind of shooting on the dark. But again, at the same point, your main focus, that how the IR is been designed. So you are not comparing a point-to-point in vitro evaluations when you are talking about IR, but maybe you are targeting an extent of release to see at what time point you see 100% release for your product versus the brand, and then try to again come with some innovative comparison tool where a standard in vitro dissolution is not relevant because, again, as you said, the comparator has been shifted totally. You are comparing your ER against an IR. However, the benchmark still remains. As per the literature data, hypothetically, you need to design your in vitro and simulate so that you can get the similar in vivo profile. Thank you.
My question is for Dr. Zhang. In the FDA approach for subcategory 2 and 3, there was no mention of foreign competitors, even in the situation where the US RLD sponsor continues to market the RLD in the ex-US category. So, and also there's no RS designated by FDA. Has FDA considered whether a foreign marketed version of the RLD could be used as an alternate comparator? If so, what type of bridging information would FDA expect to establish linkage between the US RLD and the foreign sourced product? Thank you.
You hear me? Okay, sorry. Appreciate your raising up such a very debatable question. It's a more policy relevant, but however, as you saw in this session, we have so many speakers talk about the agency's effort to try to find alternative B approach. So for foreign ROD, it's we welcome you bring your proposal, and we do open the door for discussion. But at this a session, I couldn't see what's out. I mean, basically, officially, we still have to recommend you use the ROD basically approved by US FDA. For foreign ROD, there are some situations like the same company market the same product in both US and Canada. So it's a situational depending. So we welcome you bring your proposal and talk to the agency for more exploration. Thank you.
And to further emphasize all the approaches we discussed today, bioequivalence is still established on the US reference listed drugs. So although we are considering use of intermediary products or alternative comparators, your end bioequivalence is still determined on the reference listed drug via a variety of approaches as highlighted in our talks. With statistical bridging or biowaiver approaches. Great question. Thank you. Okay.
So quick question. For products that are in limited distribution, meaning there's not very much of any individual lot available in the market, has there been consideration of pulling samples for multiple lots to use as both the in vivo and in vitro comparator?
I can share some of my thoughts. I think we do see some literature discussing some products may have the lot-to-lot variabilities and it may impact BE determinations. I think if it may be case by case. So if you have such a product, you may want to share with us what would be your proposal in terms of analyzing BE.
Sorry, it's an academic question. Since Dr. Boyce mentioned that this is a cutting-edge target. So, Dr. Brooke, do you see potential for the use of neural OD or generative models for your reference bank construction?
Hi, yes, I think that for the reference bank construction, it's definitely reasonable to look at things like we've already seen a lot of approaches talking about like PBPK or those style of mechanistic models to look at BE. I think that it would have to be validated, like those models would have to be validated against the available data that we do have to make sure that the profiles reproduced are comparable. But I think that that's a very reasonable consideration for some of these reference bank constructions when there aren't alternative methods.
Do we have any questions online?
I have one more question related to the same topic. Does this workflow that was presented by Dr. Langevin, sorry for the pronunciation, does it include execution of any kind of PK study with the test product, and in which phases? It was not specifically mentioned where this would come in, like.
Hi, yes. I'm sorry that should have been maybe a little bit more clear. In all these cases, the test product would be studied. So the idea is the test study would be conducted. It's when kind of how to take the test study data and the virtual reference bank and how to do the BE comparisons to ensure that you're not inflating error or introducing bias with these alternative reference approaches, and that the virtual comparison maintains validity. But the test product would have to be studied, and from what we suspect, the design of that study also may impact some of the considerations in terms of what you would see as a valid methodology. So that is something that has to be taken into consideration.
Hey, I have one final question before we close. So we had a lot of discussion about these strategies being written down in our product-specific guidances for specific products. So my question towards MJ and then others who want to chime in is, what are the other efforts that the agency plans on doing to expand these approaches in product-specific guidances? And will there be efforts to revise the PSGs as these products are approved using alternative approaches?
Yeah, so one of the presenters talked about how this utilizing this alternative approach for solid oral dosage form has been really useful and all. So moving forward, we are trying to utilize more of this reflected in our product-specific guidances. I really like to kind of deviate from the so-called traditional approach. So I really welcome our group revising and reflecting our new way of thinking in our guidances. So you will see more of these in our guidances moving forward. That I can guarantee you. But because many of our FDA presenters had this disclaimer, I do want to talk about, just briefly going back to this foreign competitor. I'm putting a disclaimer. It's my personal opinion. I feel like we have a room to be open-minded when it comes to using different products available. And because whenever the term comes up, the foreign product, then within the agency, especially the policy, maybe they get a little concerned because this is not just the scientific aspect. But if one can consider this approach from scientific bridging, and then if you can utilize all available information to indicate how the product that you're trying to use can be used for comparison, I feel like there is some room for being open-minded. So once again, I'm putting a disclaimer. Thank you. And thank you all for this wonderful presentation. I really learned so much from the case examples from the industry. Thank you so much. It's been really nice. Thank you so much.
I just want to add one, again, not like it also, this is, okay, disclaimer. Okay, concern. Okay, so I also review biosimilar drugs. So I just want to share one experience from biosimilar products. So if the PK similarity studies, like a three-way PK similarity studies are established, meaning that the three pairwise, like between the test versus EU RLD versus the US RLD, if the three pairwise bioequivalence similarity can be established, then in the clinical endpoint biosimilar studies, comparative biosimilar studies, the EU RLD can be used as the reference. So yeah, something to consider.
All right, so I'd like to give a thank you again for our panel here.
And I would like to welcome Dr. Sam Rainey up here to give our concluding remarks.
Thank you. Thank you to all of our presenters and panelists, and really a fantastic day. At this point, we'll say thank you and